Thank you. Okay. Thank you, Charlie, for that kind introduction and for this opportunity to present our work on plant epidermal cell walls. So today I'd like to talk about our study on plant epidermal cell wall, which is a material that is able to combine both strength and extensibility, and our efforts in trying to unraveling the mechanical and structural dynamics of this material. So as we know, unlike animals, plants don't really move around a lot, but still in nature, they are subjected to a very wide range of forces. For example, with wind, they have to deform in a whole organism scale, and as well as the tissue tension, where you can think about epidermal cell wall as a skin of the plants, that it has to undergo this overall tissue stress to hold the overall mechanical integrity of the plants. And even at the level of the single cell that has to subjected to turgor pressure to withstand this mechanical integrity. And in this case, the plant cell wall, which is the extracellular material that surrounds all the cells in plants, has to be strong enough to keep the mechanical integrity of the plants. And in the case of epidermal cell wall of growing tissue, they also need to be very extensible enough to allow plant cells to grow. So this requires overall the epidermal wall to be a material that is both strong and extensible to accommodate this multifunctional purpose for plants. And naturally, one would be curious about what's the structure of this plant cell wall where this seemingly contradictive mechanical property starts to emerge. So primary cell wall, especially epidermal cell wall, often structured as a cross-lamellar material, as shown here, a 3D model of plant cell wall, where you can see that we visualize 3 different lamella here. And if you look in each lamella, there are cellulose microfibrils preferentially aligned at different directions. And if we look into each lamella, they're mainly composed of 3 different components. We have cellulose microfibrils that's composed of crystalline and amorphous sections that carry most of the wall stress. And then we have hemicellulose, that's mainly xyloglucan, that is able to associate with cellulose microfibril to form this network. And last but not least, there's also pectin, that's highly hydrated gel that basically fill up all the spaces between cellulose bundles in this network. And although we have a relatively good understanding of what cell wall structure as well as its function should be, our understanding of this structure property and function relationship are still not very well. So in our case, we're curious and want to have a better understanding of the mechanical property of the primary cell wall that is not only important for us to understand the wall structure as well as its function and help us establish this structure property and function relationship, which will be important for the design of biomimetic materials that has similar property. And the mechanical property are also a very important input for plant models that has been recently frequently used to study plant growth as well as morphogenesis. And these mechanical property are crucial and fundamental inputs for this kind of models. So these will lead to our first question, where we want to characterize the intrinsic tensile mechanics of epidermal cell wall, starting with question like how extensible and strong are the epidermal cell wall, and what happens if we stretch the wall cyclically and repetitively, like how nature would potentially impose that forces on plants. So the system, since we want to only analyze the mechanics of cell wall, so we want to exclude any effect of living cell, turgor pressure, as well as very complex plant cell geometry. So onion epidermal wall turns out to be a great system where we are able to isolate this 7-micron thick onion epidermal peel that only consists of this sheet of single layer of outermost primary cell wall of epidermal cell. So this is simply a material that doesn't involve any living cells at all. And we are able to expose the cell wall, as you can see here, with the teeth of the side wall that has been teared apart. And this material can be easily trimmed into desirable size and standard size for mechanical tests that's amenable for our stretching device. As you can see here, we are able to stretch the onion epidermal wall uniaxially and monitor the applied force as well as displacements at the same time. So the way we quantify its mechanical properties by plotting this stress strain curve, where the stress is the applied force divided by the cross-sectional area, while the strain is the amount of deformation divided by the original sample length. And by plotting the stress strain curve, we're able to analyze and visualize basically how does the slope of the this curve changes, which is indicative of the stiffness of the wall material. And by subject the wall to different kind of loading tests, we're able to probe different kind of mechanical properties of the plant cell wall. So the first experiment we did is to stretch the unepidermal wall as much as possible onto breakage or to the limit of our instrument. And here the stress strain curve showed that the wall is able to stretch to half of its original size without breakage. And in terms of the strength, it's able to withstand strength that's more than 15 megapascal, which is comparable to the strength of a rubber band. Overall, showing that the cell wall is indeed a very strong and extensible material. And if we take a closer look at the stress strain curve and plot the slope of the curve as a function of strain, here showing the modulus over strain, we can see that the slope of the curve is not linear, that the wall stiffness actually changes at different strains, showing that overall this is a very nonlinear response, and which is indicating that the underneath molecular movements going on in the cell wall is different when we stretch the material to different strain levels. And furthermore, we to test how the reversibility as well as the response of the cell wall when we load the material and unload it and do it repetitively, how would wall respond to that? So we can see that after the first cycle, the second cycle look different from the first cycle, indicating that there are some irreversible deformation happening in the cell wall. As we can see that there are the wall only start to bear loads after this amount of irreversible deformation, which we call it plastic deformation. And it started from the second cycle, you can see the third going up to sixth cycle, they basically overlap on top of each other, indicating that starting from the second cycle, the cyclic loading behave more like elastic material, where the deformation remain mostly reversible. And one interesting thing we see during the cyclic loading is that if you look at the elastic curve, the loading curve and unloading curve follow different paths. And this phenomenon is often referred to as hysteresis, and the area that's enclosed by these two curves indicating the amount of energy that is dissipated during the process. And hysteresis is actually a very typical feature of this kind of material, where we term as viscoelastic material. So it's a kind of material that has both elastic and viscous property. So we can imagine trying to pull a parallel of springs in very thick and viscous honey. So the elastic part coming from the overall strength is still able to return to its original position as a elastic property. However, since there has to bear the resistance and accommodate the deformation of the viscous honey, so it also involves this time-dependent component coming from the viscous flow of the honey-like material. So one way to or one test that we were able to test this viscoelastic material is by stretching the material at different strain rate, where we can see that if we stretch them to the same stress, the slower we stretch it, the more time we allow it to deform, and it will reach a higher amount of deformation during that time. So with this, we're curious to see how would cell wall behave. Does it would it stretch like a viscoelastic material? So the first thing you can imagine, we similar to the analogy I just mentioned, that the cellulose microfibril, where we can almost treat it like parallel and intertwined elastic springs, stretched inside the pectin, which is the major visco component in the plant cell wall. So the first test we did is try to stretch pure pectin strips to see if the pectin is indeed a viscous material or viscoelastic material. And you can see here in this test, we pre-stretch the material 6 times to eliminate the plastic deformation, and then we change the stretching speed by 100-fold to see how the deformation would change. You can see that the slower we stretch the material, the higher amount of deformation it actually reach, suggesting that the deformation of the pectin is indeed rate dependent. However, when we repeat the experiment on the onion epidermal wall, we actually see that when we change the strain rate by 100 folds, the stress strain curve basically overlap on top of each other, suggesting that the elastic deformation of the wall actually remain rate independent, showing that by comparing these two graphs, we can it shows that the pectin the viscous flow of the pectin may play a very limited role in this overall elastic deformation of the cell wall. And in order to test if this rate-independent elastic deformation is a typical behavior of cellulose-like material, we also repeat this test on the pure cellulose composite, and we can see that indeed the cellulose composite also show this rate-independent elastic deformation. So by comparing the cell wall material with pectin and cellulose composite, it's overall suggesting that the onion epidermal wall behave more like a elastic cellulose network rather than a viscous pectin gel reinforced with cellulose. And this test also allows some interpretation in terms of the molecular movements underlying this rate dependency. And we can see that combined with our molecular insights from a recent modeling observation, where we saw that the elastic deformation mainly comes from the straightening as well as reorientation of elastic cellulose fibers. Our results suggesting that these elastic deformation require very little relaxation time, that's at least less than 2 seconds, that overall this deformation appeared to be rate independent in our experimental condition. And furthermore, we want to explore, now that we know that elastic deformation is rate independent, we want to explore if the plastic deformation also potentially be rate independent or not. So to do that, we stretch the material at different strain rate and average out the first time loading curves, which would include the plastic deformation. And we see that the wall show rate dependency only when we stretch the wall at very high speed. And we further examined to test if this is also reflected in the residual plastic strain. And we measure the plastic strain after the first loading, and we found that indeed we only see a differences in plastic strain when we stretch the cell wall at very high speed. So overall, this is showing that the plastic deformation does show a certain degree of rate dependency, but only when we stretch it very fast. And by looking at the time scale, it is indicating that it probably requires a relaxation time that's between the range from like 3 to 43 seconds. And this generates some very important molecular insights in terms that it is indicating that the plastic deformation is not likely coming from the bond breakage in the cell wall, because one would expect that the faster we stretch the material, the more bond breakage we would introduce in the material, thus more plastic deformation, which is not the case here. So combined with our observation in the modeling data, where we show that the sliding between cells and microfibril is the major contribution of the plastic deformation, these experimental results further showing that the sliding is more likely to be the mechanism for plastic deformation, as this is a motion that likely to require a certain amount of relaxation time that's probably within the range of 3 to 43 seconds. So from the monotonic loading, we already showed that the overall stress strain curve is nonlinear, and from the cyclic loading test, we see that by stretching the wall cyclically, we can analyze different kinds of mechanical property. So in order to study how these different wall mechanics evolves and change at different stress strain levels, we stretch the wall progressively and cyclically to different strain level across different phases of the cell wall. And we developed this analytical framework that is able to decompose each cyclic loading tests by different kinds of or aspects of mechanical properties that allow us to quantitatively analyze different wall mechanics at different stress and strain levels. And in interest of time, I will not get into the all the parameters here, but only point out two major observations we found. First is in the strain analysis, we see that even after the initiation of the plastic deformation, and you can see the blue curve, which is the plastic strain, start increasing, the elastic strain continues to increase after the yielding starts, showing that this extensibility of the cell wall is a result of combined elastic and plastic deformation. And what's more interesting is that as we see, starting from the phase 2, the wall starts to deform plastically. However, this doesn't really result in a decrease in the overall elastic modulus of the wall. The wall is still able to maintain its maximum elastic loading as well as unloading modulus even after extensive plastic deformation, showing that the wall is not experiencing a significant amount of loss of load-bearing component, and it is able to recover its elastic modulus even after extensive loading. So to summarize this part, from the monotonic loading, we can see that the wall is strong, extensible, with a very nonlinear stress-strain behavior. And following that, we characterize its rate dependency and found that the wall behaves more like a cellulose network capable of sliding rather than a pectin gel reinforced with cellulose. And finally, with a framework of progressive cyclic loading and analytical framework, we're able to show that the wall extend with this combined elastic and plastic deformation without compromising its stiffness. So overall, these suggest that we are able to propose this framework that is able to review the dynamics, mechanics of the wall with molecular insight and provide a tool to study the nonlinear mechanics of other wall materials. And with now that we have a relatively good understanding of the wall mechanics, we are curious to see what's the underneath molecular mechanism and structure movements that enables the wall to have these such dynamic mechanical properties. So this leads to our second question, where we're curious about the structural dynamics of epidermal wall upon tensile loadings, that we are trying to establish this structural mechanics property relationship in the primary cell wall. So to approach this question, we're trying to characterize the wall structure from different scale, starting from the microscale that we can see potentially network rearrangement, and nanoscale in terms of the ordering and spacing of wall polymers, and finally at atomistic scale to see what the tensile mechanics resulting in any changes within crystalline cellulose microfibril in terms of its length or packing. So previous study from our lab has observed some individual microfibril movements during cell wall extension where we are able to stretch the cell wall under atomic force microscopy and observe this behavior such as kinking, separation, and shearing of fibers, which is similar to the sliding I mentioned in the previous slides. And following that, we also develop a coarse grain model that is able to predict distinct movements of cellulose microfibril under uniaxial stretching, where we see the straightening and sliding of fiber lines towards the stretching direction, as well as curving and reorientation for fibers that passively accommodate to the overall dimensional change of the cell wall. So these, both of these observation is able to characterize the cell wall mechanics and the underlying molecular mechanisms, starting from the elastic region to the initiation of the plastic deformation. However, our understanding of how the cell wall polymer rearrange themselves upon very extensive loading beyond 20% still remains not well understood. So one conundrum we were having here is that based on our observation, we think sliding event is a very important mechanism for plastic deformation. And one would think that once the fibers start to slide, the contact area between fibers would become smaller and smaller, which means it should be easier for us to further stretch the cell wall, meaning less amount of stress need to be applied on the wall to induce further plastic deformation. However, if we look at the stress strain curve, we can see that even after the plastic deformation start to happen, it still require increasing amount of stress for us to further stretch the cell wall plastically. So this raises the question of what's the mechanism that's enable the cell wall to withstand more and more stress even when it starts to yield, which is a phenomenon that's often referred as strain hardening as well. So to characterize the large scale deformation of the cell wall, we used various kinds of tools to study the cell wall from mesoscale, where we can use atomic force microscopy to visualize the overall network rearrangement. Then we could follow up that with a small angle X-ray scattering to study the ordering and alignment of fibers. And finally, use wide angle X-ray scattering to probe the crystalline structure of cellulose upon deformation. And this project is in collaboration with Joshua Del Mondo from Enrique Gomez and Esther Gomez group at Penn State University, where he performed all the X-ray scattering experiment. So first, we starting from trying to look at how does the tensile deformation at large scale affect the overall dimensional change of the cell wall. So you can see here at a glance that when we stretch the wall over about like 20%, the transverse direction or the width of this peel start to decrease. And if we further quantify that at the cellular scale, where we are able to measure the quantify this dimensional change using Poisson ratio, which is the negative transverse strain over the longitudinal strain. And we can see that these measurements reach a very high level when we stretch the wall beyond 20%, suggesting a very high degree of transverse shrinkage of the cell wall. And if we further quantify how this dimensional change would affect the area of the plant cell, we can see that the area of the cells actually start to decreasing starting from the 20%, suggesting that the overall to accommodate this large scale deformation, the cell wall polymers has to undergo very large scale rearrangement. And considering that almost like 85% of the cell wall is consisted of water by volume, it is possible that during the deformation, water can be squeezed out of the cell wall network, and the matrix like pectin can be compressed during this process, where the polymer crowding could happen. And we were thinking that maybe polymer cell wall cellulose microfiber gets closer to each other and potentially forming larger bundles to resist the further sliding of the fiber. So to further examine the polymer deformation at a closer scale, we developed this method that we are able to fix the deformed cell wall structure at a deformed state that allow us to incorporate these samples into X-ray scattering and AFM measurements. So by air drying, we're able to actually fix 98 to 99% of deformation in the tensile mechanics. So by stretching and air drying the cell wall under tension, we're able to preserve the plastic and elastic deformation that allows for further examination. And by stretching the cell wall and relax it, and then air dry them, we're able to only preserve the plastic deformation and allow for further examination to basically give us insights about what kind of structure deformation is reversible and irreversible. So the first method we use is atomic force microscopy, which is able to visualize the surface cell's microfibril topography. As I mentioned before, by peeling these single layer cell walls, we are able to expose the newly deposited cell wall surface. And AFM is able to use the probe to physically touching this surface fiber and review its overall organization and arrangement. So here example of what we see in a cell wall that hasn't undergone any deformations. And we can see that the AFM is able to visualize this cross-lamellar structure, where we can see that in different lamella, there are fibers preferentially oriented at different directions. And it's able to resolve single or bundled cells microfibril pretty well. And we are curious about how would extensive deformation of the cell wall change this cross-lamellar structure, and how would fiber rearrange themselves. So first, when we observe the cell wall that stretched from 0 to 5%, we didn't really see a change in the structure. We are still, at least by eyeballing observation, and we still are able to see this cross-lamellar structure. And just to point it out that this is not, we're not looking at the same spot. We stretch the onion peels from the same onion to different degrees and dry them for measurements. So we're not looking at a continuous change of the same location. And once we look at the peels that stretch to 20 to 50%, we start to see this orientation-dependent rearrangement of the cell's microfibril. Where if you look at the top panel, this is a part of feature where we see very straight and ordered cells of microfibril aligning towards the stretching direction. And these are the fibers that is the major load-bearing component towards the stretching direction. That and on the other hand, if we look at the lower panel, we are actually able to visualize these very fibers start to curve and kink in the direction that's perpendicular to the stretching direction. And in the case of the image I here, you can actually see that the surface fiber are curved in the transverse direction, and underneath that we can see a straight and ordered fiber aligning towards the stretching direction. So overall, we observe these orientation-dependent features in the cell wall upon very large deformation. And if we look at the peels that has been stretched and relaxed, we see that we can no longer observe those very straight and ordered fibers, while there are still certain irreversible kinks and curving of the fibers in the network. So one highlight and observation I want to point out is that previously we were, before we get into this observation, we were wondering during the polymer crowding if they can form large bundles, and that's resisting the further sliding. However, if you look at the figure CDE here, we didn't really see formation of large macro bundles. Instead, we see very aligned and straight fibers that stay well separated. So this is saying that there's probably something in between cells and microfibril that's keeping them from forming these large irreversible bundles upon stretching. And as I mentioned before, with AFM, we're only probing the surface cells and microfibril. So we want to further investigate and look into the cell wall to see if this is a feature that persists through the cell wall layer and different lamella. So the approach we use is the small angle X-ray scattering. With the transmission mode, we are able to review the phase-on view of cells and microfibril and able to quantify its features of order structures and nanoscale, and most importantly, collect information throughout the whole cell wall. So first, what we see is that similar to what we observe in the AFM image, the more we stretch it, we start to see this realignment of fibers along the stretching direction as we see in the AFM images. And a very important and interesting feature we see is that in starting from 20%, if you look at image CDE, these very obvious and light dots start to form in the X-ray profile. And this is an indication of that there's a very ordered feature start to emerge when we stretch them at 20%. If we plot that in the horizontal direction, we can see more obviously that there's a peak start forming from 20%, so, which we attribute to that this could be an ordering feature that comes from the center to center packing of cells and microfibril oriented towards the stretching direction with a distance of 7.4 nanometer. So this is would combined with our observation from AFM indicating that throughout the whole cell wall, these very extremely ordered features is persistent in the whole cell wall scale and is a very, very dominant feature when we stretch the wall to the large deformation. And if we examine the sample that has been stretched and then released, and we can no longer observe these features, suggesting that similar to the AFM observation, these very ordered and aligned cells microfibril network only emerges as an elastic deformation at high strain. And overall, this is showing us evidence that the more we stretch it beyond 20%, these very ordered and aligned networks start to emerge. And following this, we also use the wide angle X-ray scattering that is able to reveal the features of ordered structure at atomistic scale. So this is basically looking at the cell wall with where we are able to visualize a much small features. For example, we're able to look into the crystalline ordering of cellulose chain within the cell's microfibril. By looking at the features like 004, we're able to tell whether the backbone of the crystalline cellulose has been extended or not. And by looking at the features like 200, we are able to tell if the spacing of the chains within the fiber has changed or not. And interestingly, by plotting different features along the strain, we can see that this 004 feature, which corresponds to a quarter of the unit cell length of the cellulose, as we stretch the wall beyond 20%, the backbone of the cellulose actually gets extended, suggesting that the cell's microfibril in the primary cell wall is able to withstand significant amount of stress that the crystalline backbone of the cellulose chains are getting extended. And on the other case, we can also see that the packing of the cellulose chain within the microfibril also decreased as we see this packing distance start to decreasing at large deformation. And we only see this increase in the backbone and the tighter packing in the chain in the longitudinally aligned fibers. That's mostly the load-bearing component of the lamella in the cell wall. And this is saying that overall, the longitudinally aligned fibers, we are able to see that the cell's backbone are extended and the chain pack more tightly within the fibers as we stretch them extensively. So to summarize this part, using a suite of characterizing tools, we're able to see from AFM that there are overall large-scale orientation-dependent rearrangement of fibers, and we see these very ordered structures of cells microfibril upon loading. And with the X-ray, small angle X-ray scattering data, we further show that these very ordered features emerges as a reversible features and with a center-to-center spacing of 7.4 nanometer. And we think that potentially there could be wall matrix like pectin or even xyloglucan that is preventing this formation or new contact between cells and microfibril upon loading. And finally, with wide angle X-ray scattering, we're able to show that the longitudinal cellulose are actually undergone very strong bearing significant amount of loadings that the backbone gets extended and chain packs more tightly. So combine all this observation, we have this hypothesis that the reason for an increased amount of stress as the wall starts to slide could potentially due to the polymer crowding, which means that the tighter the cells microfibril come closer to each other, they are able to form these compressive forces on top of each other that prevent them from further sliding, and this increased resistance for sliding then would require much higher stress for us to stretch the cell wall material. So to kind of like summarize what we observed here from the previous modeling data and as well as the X-ray scattering data, we can see that in the elastic region, we can observe the straining and reorientation of cells microfibrils. Well, in the modeling data, we can also see that at the initiation of the sliding, we are able to see the sliding between cells microfibril. And once we reach a very large deformation, we start to see this very ordered and well-aligned fiber network start to emerge, potentially due to the polymer crowding that will lead to an increased resistance sliding that then allows further stretching of the cellulose backbone that enable the cell wall to withstand higher and higher amount of stress even after additional deformation. So now that we have relatively good idea and also established this framework for the characterization of structure and mechanics of the cell wall, then we want to further explore how does this relationship could correlate to the growth ability of the plant cell wall, which then we want to apply this framework to Arabidopsis stem. So the Arabidopsis is the model system in plant, especially dicot material plants, where there are abundant resources in terms of the understanding of its biological process, as well as mutagenesis methods and mutants, where it could be a very good resources for us to study the dynamics of cell mechanics and structure to the growth ability of the cell wall. So we want to, currently right now, we're trying to explore how could different mechanical property relates to the cell wall growth, and how does the wall structure could potentially change upon growth. So previous study has shown that if we look at a stem of a Arabidopsis apical stem, that's only about like 5 to 6 centimeters tall, and where it still have tons of like growth potentials. We can see that we're able to isolate these segments and test their mechanical properties. And similar to what we did with onion epidermal cell wall, we're able to show that the apical Arabidopsis stem shows similar stress response in terms of the monotonic loading, as well as rate-independent stress-strain deformation. Overall, showing that we are able to transfer this mechanical property characterization method to Arabidopsis apical system as well. So also previous study have shown that with the it's kind of like a phenotyping of the growth ability, where we are able to see the further away, if you look at further away from the apical stem, the slower they actually grow. So this is as shown here, that the top, and which means the younger segments, actually have a higher growth rate. And if we want to only characterize the material property, we can here, as we shown here, we would exercise the different segments, and then did a in vitro creep test on these different segments, where we subject these segments under constant loading, and we activate the activity of endogenous cell wall loosening protein expansing. And we can see that in vitro response that the younger stem has a much higher creep rate compared with older stem overall, showing that the younger stem have a stronger growth ability as well as in vitro extensibility. And the next step that we are also currently doing is to see if we can find any correlation between wall mechanics to these almost 3 times differences in the growth rate as well as extension rate. So currently our preliminary data here shows that if we subject the Arabidopsis apical stem to progressive cyclic loading tests, as we can see here, this curve show that the black curve, which is the first segment, the youngest segment, show the younger stem actually appear much softer compared with older stem. And we can also see that by plotting the elastic unloading modulus as a strain, where we can see this almost 5-fold differences that is somewhat similar to what we see in the growth rate. And what's very interesting is that when we plot the elastic strain of these different segments, there appears to that the ratio of reversible and irreversible deformation stay the same for different segments. So this is suggesting that the underneath molecular movements and their reversibility are the same throughout different segments. And the differences in the stress mainly comes from how difficult and how much stress it requires to induce this same amount of deformation. So there are a couple of hypotheses we can draw. Is potentially comes from that the basal stem might have higher amount of cell wall and potentially have more cells microfiber oriented towards the growth direction that's limiting the growth. So combined, following that line of thinking, we also have previous evidence showing that surprisingly, when we measure the biomass of the cell wall material across different segments, we found that there's really no change observed in the biomass of those cell wall as well as the cross-sectional area of the cell wall material. And we have additional evidence from solid state NMR where we found that there are modest change in the cell's content. So this is saying that these differences in the elastic modulus and stiffness does not come from like how much cell wall material you have. Then in this paper, they also did this test. They observed different cell wall using small angle neutron scattering, and they didn't really observe differences in terms of the ordering and bundling and of the cell's microfibril. So then this leaves us with one potential hypothesis is that given the same amount of cellulose, one factor you can play with is how much cellulose is oriented in the stretching direction or not. So it is possible that with the younger stem, there are more higher percentage of fibers oriented transversely that you can allow more growth in the vertical direction. While for the basal stem or older stem, there could be more cellulose microfiber oriented towards the stretching direction that's resisting the growth. So with that, this is a study we're still currently working on. We are trying to, basically the goal is to study the correlation between this wall mechanics and growth ability, and if it has anything to do with the cell wall orientation, and to see if this correlation persists in different kind of cell wall mutants or modified cell walls. And for a final summary, so first we start with a mechanical test where we want to characterize the intrinsic mechanics of cell wall and how dynamic what their dynamic looks like at different stress and strain, and then we try to establish this structure mechanics relationship by looking at the cell wall rearrangement at different scales, and then finally, and currently we're trying to look at what's the structure and mechanics growth relationship. That is it possible that we can find any correlation between the structure and different wall mechanics like elastic modulus and see how they could potentially relate to this growth of the cell wall and how much of this material property of the cell wall would affect the growth. And finally, I'd like to, for acknowledgement for our the guidance and support of my advisor, Dr. Cosgrove, as well as our current lab members for their technical support and all the help, and our collaborator, Dr. Joshua and Dr. Henry Gomez and Nestor Gomez for their help with the X-ray scattering measurement and our funding resources. And that's all I have, and I'm happy to take any questions. Thank you.
Great. Thanks so much, Jingyi. So it looks like we have a question from Becky Wells. Becky, if you want to just say your question out loud, go ahead.
Yeah, so I just have to say that was just a beautiful, beautiful talk. I mean, just incredible data. It was just amazing. I had a question about the backbone stretching that you described in part 2 of the talk. And I wonder, so is that backbone stretching, is that actually reversible or irreversible? And do you see the same thing in the Arabidopsis stem at all different in all different segments?
Oh, so that is a very good question, which is also a part that I am curious. So unfortunately, I think with our measurements, we did the reversible and irreversible example in the small angle X-ray scattering and AFM, but we haven't taken a closer look at that features in the wide angle X-ray scattering. So I would definitely try to look at those data again and to see if that feature is reversible or not. And because we would want the backbone stretching to be a reversible feature for sure.
Okay.
Yeah. And for the second question, which if I understand correctly, you're referring to see if we see any backbone changes in the different gradient of growth.
Exactly. Yep. Mm-hmm.
stand, and we haven't done that test. I would say this is also down the line where we want to potentially isolate the epidermal cell wall or simply a Rhabdopolis apical stem and look them with the X-ray scattering and see if we can observe any, like I said, if there are any orientation preference in different segments, as well as potentially the differences in the packing and backbone and chain packing in different segments. So that's definitely something down the line that we are interested in.
Yeah, it's very interesting. I'm going to have to think about this for a long time to wrap my head around it. But, thinking about the animal ECM, and, you can beat the analogy to death a little bit too much, but I think that there are some structural similarities, at least initially. And yet with significant stress, the changes are plastic. And it makes me wonder what some of the structural differences are in the big biopolymers that might lead to the differences in behavior between plants and animals. So,
yeah.
but I have to think about it.
Yeah, it's fascinating. Also, like we're tackled with this problem, although we have a hypothesis of like what potentially the mechanism for this string hardening, but we still need more like tests to see like removal of certain polymers to see if we can change the like slope of this linear region or what. But yeah.
Yeah. Well, really nice work. Thanks.
Thank you for the suggestion.
So Jingyi, I'm going to ask you a question about pectins, which maybe is not so surprising. So in the onion cell wall, there's a lot of cellulose and also a lot of pectin. And do you see any evidence in your experiments, either AFM, SAX, or wax, for any reorientation of the pectin network under strain? I mean, it's a little bit harder to see, I think, than cellulose with some of those methods, but you might imagine that the pectin either gets dragged along with the cellulose, or that it's maybe getting folded up in interesting ways, like those perpendicular cellulose microfibrils that you imaged.
Yeah, so I think there's actually so starting with our experiment in the AFM measurement, we won't be able to like visualize the pectin because we actually try to remove the surface pectin when we are trying to visualize the cellulose, because when we dry the material, the pectin gets really dried and we can only visualize that like porous network of the pectin, and that's blocking the cellulose. So for the AFM, we won't be able to see the pectin. And in terms of the SAX and wax data here, I think we have a very low, I would say, contrast. The contrast mainly come from the cellulose and pectin, so we won't be able to tell pectin. But with that question from other like potentially work, I think there has been some previous work from FTIR where they already show that there are some realignment of pectin when we stretch the cell wall, although not probably at large deformation. But I also did some experiments with the Raman microscopy where I stretched the cell wall material under the Raman microscopy, and I was able to see that we see a reorientation of the pectin aligning towards the stretching direction as we stretching the cell wall. So that's definitely true, that it's not just cellulose that's reorienting, other polymers reorient as well.
Yeah, and it'd be interesting to actually intentionally dry the material and look at the pectin network and see if it's changing, or to treat with calcium or something like that to, make the pectin network stiffer for AFM imaging. So those would be fun experiments to try.
Yeah.
Not that you have an infinite time in your PhD to do it, but yeah.
Yeah, that's actually would be an easier experiment because we don't have to digest away all the pectin layers. So
exactly, just skip that processing step. So
right. Yeah. Thank you. That would be cool to see.
Any other questions in the audience? Especially encourage any students or postdocs to ask questions. Well, maybe I'll sneak in one more question, Jingyi, which is having to do with the STEM studies, which are really nice and interesting. What are your plans in terms of mutants, right? Because some of the mutants that we've characterized in the Center for Lignocellular Structure and Formation have differences in stem growth rate and diameter and things like that. So do you have plans to repeat any of your mechanical testing on mutants that lack, for instance, zyglucan or different forms of pectin or are defective in cellulose production?
Yeah, that's a great question. We actually already grown a bunch of like X61 and 2 mutants, which lacks like xyloglucan, and I already harvest all the stems that's ready for mechanical test. And actively trying to like talk to different groups as well, just to see if we can get like cellulose or pectin mutants to see how they would respond mechanically. The only question is how we interpret it, because the mutants is a lot more other factors should be considered in that way. So definitely will be interesting as a future work.
Yeah, very cool. One of our mutants that we've characterized called PGX2 activation tag actually bolts early. It produces an inflorescent stem early, and it generates a thinner stem that seems to lignify prematurely. So that one might be an interesting model to do some mechanical analysis of. We've done a little bit with sort of bending resistance, but not in the longitudinal growth direction. So
yeah, sure,
we
that
can
would
talk
be
more if you like. Yeah.
Yeah, of course. Thank you.
Any final questions from the audience? So if there aren't any further questions, I would encourage you to reach out to Jingyi if you'd like to get in touch after the fact. And you can also contact Annie Zhang. She can put you in touch with Jingyi if you don't have her email. But again, a big thanks to Jingyi for presenting in our seminar series today. It was a wonderful presentation. I'll echo some of Becky's compliments on the quality of the data and the quality of how it was presented. And looking forward to seeing you all at the next iteration of the Future Leaders in Mechanobiology seminar series, which will be on December 13th. Have a great day, everybody.
Thank you. Have a great time.
You can hang out here. I made you co-host, so even if I leave, like the meetings still stay on. I'm gonna stop recording though.
Okay, sure.